Joe, I've got it covered. Thanks. Good afternoon. We're honored to have today Dr. Pooja Shivshankar visit us for the final grand rounds of September. Remember that next week at the San Antonio Airport Hilton is the 2012 Conference on Aging. If you haven't signed up, now's the time. She comes from medicine's cardiology department here at UTHSCSA, where she's a research scientist. Dr. Shivshankar is currently working on the molecular aspect of aging lungs using mouse models. Her experiences focus on the fields of senescent lungs, dysregulated inflammation, pulmonary fibrosis, and pneumococcal pneumonia. Dr. Shivshankar has long-term interest in studying the role of oppressive aging factors and increased vulnerability to age-associated pulmonary and cardiovascular diseases, and how these adversities predispose the elderly to infectious assaults. These molecular aspects, along with the update on diagnosis and possible improvements in preventative measures and treatment strategies, will be highlighted in Pooja's presentation today, which is entitled Molecular Aspects of Cell Senescence in Age-Related Lung Diseases. Thank you, and take it away, Dr. Shivshankar.
So, now currently I'm working on senescence and other age-related oppressive factors that affect lung mechanics in the elderly. Of course, we do have patient samples from COPDs and IPF patients, but mostly our research is based on animal models. So, and those are mouse models, and we try to simulate the conditions of smokers by inducing oxidative stress, and idiopathic pulmonary fibrosis by inducing DNA damage through bleomycin, which is an anti-cancer drug. And it's a standard tool to induce fibrosis, and it is supposed to cause, as a side effect, pulmonary fibrosis in humans too. So, taking advantage of those two systems, we have studied the senescence and the mechanism of inflammation, which can contribute to infectious diseases. And the model we have here is Streptococcus pneumoniae. And last year I presented the whole data set from the paper which we published in Aging Cell. And today I'm going to talk to you a brief account on that, and then how we have progressed after that. Although I don't have much data from the present study, but still I'm going to convince you with our recent findings. And maybe I'll share with you the diagnostics and the treatment strategies which you should be aware of, and you can also help me understand them. Thank you. So, to give the disclaimer, I have nothing to declare or disclose and no financial interest. There are many vaccines and other products which I'm going to name in this presentation, but I'm not associated with them. So please take it as only through the educational point of view. So age-related diseases, anybody, if you ask, if you age, what is that? Whether it is a healthy aging or you have disorders which can lead to disease. So that's a question always people have. But biological theories will make you understand what goes wrong with the cells or with the system or the organ function that can lead to aging. And those are DNA damage theories, and DNA damage theories, telomere theory, reproductive cycle theory, cross-linkage theory, and free radical theory. If I have to give you a simple definition, DNA damage is due to any UV radiation or some sunburn or some chemicals which can cause DNA damage into your cells. They can get intercalated into your DNA, and they can impair your DNA replication. And the telomere theory, which is really emerging in the field of aging, because every DNA molecule is bound as a chromosome, and those chromosome ends are preserved by the telomeres. Those are short-term repeat sequences which preserve the exons and the functional protein genes. So, and the telomere length is maintained basically by telomerase enzyme. And it has been shown that as you age, you have a decline in the function of telomerase activity, and that leads to chewing of these telomeric ends. So now your DNA is prone to get damaged and lead to stress. And reproductive cycle, I don't know whether it is a debatable concept or it's accepted that even females, when you reproduce a lot, you accelerate aging. I don't think, because females have a long life than men. So I have to study on that. Cross-linkage theory, this is another really interesting theory. It makes advanced glycation end products. What happens is, when you intake your food is going to get metabolized into proteins and lipids, and you have some functional proteins which are synthesized because of those amino acids which serve as the substrates. And those amino acids, when they are formed as the peptides, you have to have post-translational modifications. And that time you have the glucose residues and other glycosylases which are going to glycosylate your proteins and lipids to make them actively functional. Okay, so, but this is glycation, which is a chaotic attachment of glucose moieties to proteins and lipids. Wherever it can, it goes and attaches. And it also happens during cooking. So a kind of a preventive measure now is to check the food products where you have increased amount of ages. So if you don't have the intake of ages, you don't accelerate aging. So there is a lot of mechanisms studied behind that, but I don't want to go into details. And the last one is the free radical theory. This is the most important one, because free radicals are generated from any source. So even if you say DNA damage, telomerase activity, you have that free radical response inside. And then reactive oxygen species. Chronic oxidative stress is the leading cause of this free radical theory development. And so what are all these causes? Until now, nobody knows why we are aging and why aging is that inevitable. Can we stop? Although we have very good models from progeria syndrome as well as from Werner syndrome. And both these models refer to the phenomenon of senescence, that is, the cells age faster. And in the case of both these diseases, the patients don't live more than like 15 years of their age. Within 13 years, they should have one cardiac event. And this guy, he is a fifth grader in California, and he is 40 years old by his system function, whereas he is just 10 years old. So it is 4 times accelerated than what you normally see. And yes. Okay. Any questions? Okay.
I think there's a movie actor.
Yeah. Okay. So aging-related diseases, if I have to say now again, can I debate on if aging by itself is a disease? There are some scientists, they categorized aging in the disease category. I have seen presentations where they say diseases, aging and cancer. So aging can be considered as a disease because when you're young, you live with your comfort. And as you age, you are going to have the discomfort. So they categorize aging as disease. It's really controversial. But what I want to do is, I want to tap the disease condition of aging into 2 points. One is lifespan and the other one is health span. We have a multi-centric project going on now with Bashap Institute on these health span and lifespan studies with using mouse models. And a lifespan is generally how long you can live, no matter whether you're sick or healthy. But a health span is the one which governs that lifespan. For example, if you are healthy, it has been statistically shown that if you are healthy until 65 years of your age, you will cross 80. Don't be afraid. If you are not healthy by your 65 years of age, you cannot cross 69. So, because every decade of your life, you're adding one disorder in your body. So, when 1,000 things happen, only one thing emerges phenotypically. So if you're 40, you already have 4 disorders, discomforts in your body. So how severe those are, and how the 5th one is going to impact on those 4 conditions, and how by 60 you're going to handle those 5 disorders together is a big question. And those who reach 100, that means they're having 10 disorders. So even if you sneeze, if your boss is like 80 years, you'll get a holiday on that day, because the next day he'll be on ventilation. So it's a danger. And in terms of disease, if you want to see, we always say we have chronic inflammatory diseases like obesity, diabetes, and different conditions. And we have cardiovascular diseases, definitely. And pulmonary diseases, which is idiopathic pulmonary fibrosis and obstructive disease. And I just took one example of autoimmune disorder, which is arthritis. And that's my interest, pneumonia, infectious disease. So I'm going to tell you the impact of all these diseases or the disease conditions, how they can predispose to infectious assaults. And is it the danger? Or where is the danger? The danger is here. We have almost 40 million elderly at the age of 65 and over. And by 2050, we are going to have double. It is because we have very good management of the disease. We are caring our elderly individuals, and their life expectancy is indeed increased. And can we save them after 65 is a big question. So we have to see what preventive measures we can take, not only the management of the disease or their conditions, but you have to prevent them from developing any further adversities. That's my point. And community-acquired pneumonia is the major leading cause of death among the elderly. And if you see this chart, you have worldwide the incidence of community-acquired pneumonia caused by Streptococcus pneumonia ranks first, followed by other pathogens. So that's really a serious condition. And why do you think it's a serious condition? Because if you see this normal lung, it is clear. And this is a patient who is 70 years old, and he is admitted with this Streptococcus pneumonia infection. Sorry. Streptococcus pneumonia infection. So these are the early hours of infection, and his lungs are cleared while he was admitted. So they say, okay, no problem, he's fine. And they're really taking easily everything, and they want to proceed through treatment. Yes, they want to. But within 12 hours, he has acute inflammation, and within 20 hours, he dies. And why is that so? If you see the sequence of invasive pneumococcal disease, the bacteria which colonizes into the nasopharynx can get transmitted upon aspiration. It's very easy for the elderly. That's it. It goes into the sterile site. And it goes and causes pneumonia. And here, it takes normally at least 3 days to clear, normally, into a young adult. But in the case of the elderly, it disseminates faster. Within 12 hours, it's in the bloodstream. So you have a septic shock. And within 2 more hours, what is going to happen is meningitis. So most of our animals, if we give a dose to the elderly, which is equivalent to the young animals, those old animals within 2 hours, if you hold their tail, you can see them spitting. They have meningitis already. So whereas the dose is very, very little. It clears in 7 days actually. So that little dose also can cause meningitis in the aged animals. And they're not that old. According to our lifespan studies, we have even 36 months old mice, whereas the mice which we use were 22 months. So it depends how you define aging in different strains. And for this strain, it was only 22 months maximum where we can handle them, and after that, they have so many other things. You know. So this is the scenario which is really risky. So whatsoever be the conditions or age-related pathologies like obstructive disease or fibrosis, systemic inflammation by itself is a major risk factor for community-acquired pneumonia. In the study, it's a very neat study. He included patients with smoking habits, and he included patients with no smoking but have obstruction in their lung function. And he took diabetes patients, and then he took cardiovascular disease patients, and he just measured the circulatory levels of IL-6 and TNF-alpha, the two major pro-inflammatory cytokines. And what he showed, and what is my interest, is the FEV1 going down 50%, because it's just the FEV is the forced expiratory volume, that's the capacity of your lungs, how much air you can see. And if it is below 50%, that means your lungs are not functioning normally. You may know something better in terms of justification, but that's my interest. So I wanted to see what is happening in these animals. Moreover, why do we have to be so concerned with these infectious assaults? Do we have treatments? Yes, we have treatments for the elderly as well as for the children. But the organism evolves so fast that it develops into different serotypes. And these serotypes are based on the capsular polysaccharides that are covered around that organism. So the capsular polysaccharide capsule is the shield for avoiding the innate immune response and invading the cells, and it takes all these strategies based on this capsule. And luckily, based on these capsule differences, they have determined 100 different serotypes. And the vaccines which we have for children, we have PCV7 and Prevnar 13. That means you have 13 serotypes covered. And for the elderly, we have Pneumovax 23. So we have 23 serotypes covered. But imagine the danger here, 100 minus 23, that is over 70 different strains are just happily there, and they can cause the disease. And once the infection is established, the treatment you have all is antibiotics, and for that also even you have resistance to the beta-lactams in several of those strains. And you have fluoroquinolones and macrolides. We are doing a study with macrolides on pulmonary fibrosis. My interest is to see in future. So the two age-associated pathologies of the lung are IPF and COPD. And if you look at the pathogenesis of these two diseases, idiopathic is you don't know the actual cause of the disease. So you have genetics, aging, and environment. And the type of damage you are going to get here, the response is the telomere shortening. So it's a DNA damage response, which can lead to senescence and apoptosis, and that triggers subsequently mesenchymal, epithelial mesenchymal transition towards the development of fibrosis. And in the case of COPD, you have the causative factors as, again, debatable on aging, but smoking. And the emphysematous lesions have senescence, which inhibit proliferation and also downregulate apoptosis. So there's one common thing which is present in both IPF and COPD is senescence. So why don't we look at that senescence aspect, and which we know for a long time through progeria as well as Werner syndrome, what is the cause of accelerated aging. So we wanted to look at the senescence aspect in the aged animals. To give a little background on senescence, it is the phenomenon where the cells stop replicating. So just like after 60 doubling times in the in vitro culture, those cells retire, like our grandparents, they retire after 60 years. But they don't sit quietly. How our parents, grandparents and us, they also have a altered protein profile, and they keep secreting those profiles, and they change the milieu, extracellular milieu, and that milieu impacts the normal neighboring cells. And those cells, we call them as pre-senescent. Why, I will show you later. So, and that limit of the cell proliferation is called Hefflick limit. But we do not know how many times a cell can proliferate in vivo, but at some point it also reaches that Hefflick limit. So the pathway of senescence for a long time, it has been established, and it has been put into two simple major signaling pathways. One is the telomere dysfunction and oncogenes, which can lead to DNA damage response, activating P53, a tumor suppressor gene, because once you have the DNA damage response, your cells should not undergo malignant transformation. So tumor suppressor P53 comes up and it activates P21, leading to senescence. But the back part of this pathway is it's a reversible process, because anytime these cells can revert back to normal, either normal, or they can undergo apoptosis, or they can even turn into malignantly transformed cells. Whereas the second pathway is mostly related to aging, because it's due to chronic oxidative stress, and it is also induced by oncogenic signals. And the pathway has P16 and PRP, that is the retinoblastoma protein, another tumor suppressor protein that can lead to senescence. But the good thing about this senescence is those senescence cells are irreversible. They just have to wait, and with their secretory profile, the chemokines and the cytokines, they attract phagocytes and they actually get cleared. So, which is good. But if they're sitting there, there is an exhaustion of tissue repair mechanism. So they're not moving from that place, and the tissue is not repaired completely. So there is an abnormal function. And you also have aging and tumor promotion, definitely. So it's a paradoxic phenomenon. So it was very hard to accept how senescence can be taken into a good therapeutic implementations, because you cannot use senescence itself as, if you cannot make cancerous cells into senescence, because they're going to produce so much of pro-inflammatory cytokines and matrix metalloproteinases that it will increase damage of the vasculature as well as the tissue morphology and the function. The paper, the data which I discussed last year is completely in this picture. So we have normal, of course everything was on mouse model, normal aging animals, and we mimicked the COPD smoking condition by giving hydrogen peroxide in water. Hydrogen peroxide can cause generalized oxidative stress in your body. And as I told you, we used the standard tool, that is bleomycin, to induce DNA damage response. And we saw that the cells were senescent in the lungs, and they had these receptors to which this pneumococcus bind to invade the cells. And the keratin 10 is one. And the beauty of keratin 10 is it's preferentially present on lung cells, and it binds to very novel pathogenic ligand, pneumococcal ligand. And there is laminin receptor, which is present on epithelial as well as endothelial cells, and it can help the bacteria cross the blood-brain barrier. And PAF receptor is the one which can lead this bacteria to pass through the endothelial cells. And we have increased pneumococcal binding, and we have increased inflammation. So these mediators, which we term as senescence-associated secretory phenotype. So these mediators can cause a pre-senescent atmosphere in the cells, neighboring cells, and increase the bacterial binding in these normal cells. And how they do is exclusively through PAF receptor expression. And we have the mechanism behind that, which I don't want to go through. And of course, they have increased inflammation also.
Pooja, how long did you give this? You gave it in the drinking water?
Yes, for 15 days.
15
Mhm. So, what do we expect with progressive aging now? At least with our animal model, what we are going to see is if in the young we have the receptors in the nasopharynx, because I told you keratin is exclusively in lung cells and endothelial cells. PAF receptor and PIGR receptor are the ones which are going to help the bacteria colonize in their upper respiratory tract. And then once the bacteria is transmitted, you have keratin 10 to help the binding and invasion along with PAF receptor and PIGR. And then you have laminin receptor in the endothelial cells, which can help for dissemination and blood-brain barrier. Crossing the blood-brain barrier. But in the aged, the scene is very different. You have all these receptors elevated. So you have, there is an increased frequency of the bacterial binding to the lungs in the aged animals, and therefore faster dissemination, and that's the way how they succumb to death. And this doesn't end here. These receptors are co-opted by even Staph aureus. For example, in keratin 10 is co-opted by Staph aureus as well as group B streptococcus. And laminin receptor and PAF receptor are co-opted by Haemophilus influenza and Neisseria meningitis. We have done some experiments on these receptors, and there's just a kind of comparative study for our papers. So you can see that. So if the binding is more, how about the host immune response? We recently worked on the toll-like receptors. Toll-like receptors are the pathogen recognition molecules on the cells, and they are the one which elicit the immune response. And what we saw was, I don't want to show the bar diagrams and everything, and what we saw was, it was the TLR2 which helps bacteria to bind to the macrophages and elicit the immune response. And we also showed that the elderly have increased toll-like receptor. Then it doesn't correlate with what we see. We have increased incidence of the disease, and at the same time we have that our immune system is guarding towards that increased infection. Is that true? We moved on to see what happens at the cellular level. We saw that macrophages are very high in aged animals, normal aged animals. When we infect them, we have all the more significantly increased macrophages as well as PMNs. That means you have more macrophages, and TLR function is also there, and you have more neutrophils. What are they doing? They're not clearing the infection. You have increased vascular leakage. So that shows that your system is really fighting against this infection by bringing more and more oxygen through the blood. And your vasculature is dilated now. So that means those cells are devoid of oxygen. So they're not happy. Although you have number of cells, but they cannot perform. So there is the function of those macrophages are poor. That means the term is immune senescence. So those cells are also senescent, but that's our grant. I cannot tell you anything about that. So that's my grant preliminary data. I want to show why PMNs are defective. If you can see the red, I don't know. If you can see the red, which is the neutrophils, and you can see the green, which is a fluorescently labeled bacteria. Okay. In the young, after hydrogen peroxide, if I give for 48 hours, the bacteria is cleared nicely in the young animals. Whereas, although we have the same amount of neutrophils, but the clearance of the bacteria is delayed, which shows that neutrophils are not functioning. And there is a very interesting, there was an interesting talk last year how the mobility of these PMN is affected during aging, and how they cannot reach the pathogens or the toxins at the time which is equivalent to the young couples. So yeah, that's what I'm studying, and that's my ROM proposal. And to put the things in a simpler manner, we do see increased ligands through senescence, and we have increased bacterial binding because of the primary status. And once you have the infection established, the resolution is faster in the young animals, whereas the old animals have a poor toll-like receptor function and immune response, and that's the reason why they succumb to death. And an overview is bacteria is now taking all the advantages, not only in young, old people, even in young, it finds some way to enter into the sterile sites and cause the disease. And it can produce its own hydrogen peroxide to kill other microbes in the nasopharyngeal colonization. So it takes all the precautions to form that colony or to make the bacteria survive in the form of a biofilm. That's the concept of Streptococcus pneumoniae. You have the bacteria in the form of biofilms, and what you see in your otitis media in children is also a biofilm. And in the elderly, another emerging disease is these bacteria can leak from the lungs into the extrapleural space through abscesses and form these biofilms. And that disease condition is called empyema. So that's another project we are looking at recently. And so when this bacteria has strategically winning us, all we give is a nice harboring atmosphere, inflammation, and good amount of receptors to bind to so that it can kill us. And so whose situation is that? It's a win-win situation to the bacteria. We have to control that. And how we are going to control? We have to control at the phagocytosis level, we have to control at the immunity level, and we have to control at the, I would say this one will be the first, the susceptibility by itself, how we can prevent ourselves. So to do that, the diagnostics, if I have to be in the translational medicines, what I would do is every individual after 55 years, they have to go through at least 6 months once lung mechanics, and then fibrosis, parameters, and senescence. You have to include senescence because it is going to cover a wide range of diseases, including arthritis, including diabetes, including obesity, including liver disorders, many different things. If you can take 2 markers of senescence in your lab analysis. And then at cellular level, what you can do is a collagen deposition. With many organs undergo fibrosis upon aging, kidneys, and remodeling of the heart due to one attack with myocardial infarction or atherosclerosis, and pulmonary fibrosis, of course. So the measure which it is coming up now, we have done some work on this hydroxyproline and pyridine, which are the degradative products of collagen. So it can be measured in the urine. So it's a very good tool for knowing how collagen or the fibrosis, the extent of fibrosis in a human body. And of course, inflammation. We definitely need every 6 months to check what is the level of inflammation. Not only in arthritis patients, but in all other patients, we have to look at the inflammatory. It's an expensive tool, but we have to come up with some other alternative to measure. There are now bioplex systems where you can measure like 25 cytokines, 30 cytokines in one run. So it's very cheap for our research purpose we use. Instead of using $500 one kit, we can use this one assay. In $500, 25 cytokines done. And it's very sensitive, very nice, and I think clinical labs have a flow cytometry units, and that could be a very good option for us to help our elderly. And then the paracrine effect, of course. The very important thing about Streptococcus pneumoniae is even in the joints of arthritic patients, you have cultures of Streptococcus pneumoniae. You have leishmania, you have streptococcus pneumoniae, and you also have tuberculosis. You can imagine how this blood dissemination can take that organism to distal parts of your body. So, and that's the reason I highlighted K10 in the endothelial cells. There's a very recent paper on Science. They showed that keratin 10 is released from those breached endothelial layers, and those fragments are now considered as auto-antigenic, and autoantibodies are produced against your own keratin 10 that can also lead to increased arthritis and inflammation. And we have to, I want to take keratin 10 to a different level, because there are many other papers which are coming now on ichthyosis and skin disorders. That is another very rare genetic condition. So it's really exciting how keratin 10 can do things. It was known only for keratinocytes. It said, okay, you have keratinocytes. When we found keratin 10 on the lungs, we thought it's contamination from our hands. We just threw those tubes and we said, oh, we're not looking at this, maybe it's a contamination. After 5 months, we got a paper from Agerelated group, Streptococcus agerelated. They showed those mucosal layers of the uterine cervix. They have keratin 10 and keratin 4, which can mediate attachment of these serine-rich lipid proteins, bacterial proteins. And then we said, oh gosh, that was not a false data. It was true. So we started working with immunoprecipitation, and we ran a lot of high throughput technology experiments, and we found out that it was indeed keratin 10. So keratin 10 now, Judith Campisi, who's the pioneer of senescence work, she is working on keratin 10 to control mammary tumor metastasis. So that's another very exciting research on keratin 10. So we'll see how we can control. And I hope in lung mechanics we have the diagnosis of FEV. And I also hope that we have resistance and elastance and compliance. These three should be included in the lung function analysis, because resistance is the airflow that will show how consolidated your lungs are, whether inhalation or exhalation. And you have elastance. Elastance is when you exhale, your lungs have to recoil to come to the normal architecture. But in the case of emphysema and other lung obstructive diseases, what happens is these lungs collapse extensively. But by nature, there is a mechanism called compliance, which is a reciprocal of elastance. It can inhibit that extra recoiling. So if you have compliance, reduced compliance, then that means lungs have collapsed already. So if we can include this, and that's a sign of both IPF and COPD, because there are many people who are non-smokers but still they have obstructive disease. So this could be a very good parameter to include. We have this parameter for our animals. We have an instrument for animals also. I don't know how the instrumentation is for the human studies. So I kind of like this idea how we can control them. And the adversities which we have, or comorbidities which we have with age-associated lung disorders like IPF and COPD is commonly, very commonly, cardiovascular diseases. Cardiovascular diseases, I want to say that that's another thing which you have to look at, what is the condition of their heart function, because the cell wall of the pneumococcus, once it is disintegrated, those cell wall components can go into the bloodstream and get accumulated in cardiomyocytes. Okay, so that damages cardiomyocytes and heart endothelial cells and can cause myocardial infarction. Last 2011, Dr. Martinson from VA Hospital, he gave a study, published a study on these patients that elderly patients, when they come to the hospital with cardiac adversities, they are very susceptible to hospital-acquired pneumonia. And when the patients come with community-acquired pneumonia, it was a lose-lose situation again. if they come with community-acquired pneumonia, within 9 months to 11 months, they have cardiac events. They come back with cardiac events because they have this damaged cardiomyocytes. So it's really a serious concern, and we are looking at that in terms of statin therapy. So most of these cardiovascular patients are given oral statins, which is really helpful because it lowers the cholesterol level. And the good thing about it is the pneumolysine, which is an endotoxin of Streptococcus pneumoniae, uses that cholesterol to make pores into the cell and to go and kill that cell. So when you have statin therapy, there is less option for this endotoxin to go and damage the cells. So that way there is it's again a preventive measure. Pneumococcus doesn't get the opportunity to invade the cells. And as I told you that our neutrophils and our macrophages are poor in their function because they don't have enough oxygen. And the oxygen has to come from the blood. And every molecule, every pathogen they bind to, they need 24 molecules of oxygen. So imagine how much of the blood flow has to be there for the energy supplementation. So it's very hard for the blood to cope up with the demand of oxygen with our neutrophils and immune cells in general. So if you have oxygen therapy, it's very easy. It's in general practice now, and I would say that every individual after 60 should be under oxygen therapy because it can be carried as your backpack along with your coffee mug and cell phone. Very easy, no problems at all, and you are really saving yourself. You don't have to struggle with your oxidative stress. And the next thing is anti-inflammatory drugs. There's one drug which is really, really promising is the IL-1 receptor antagonist. IL-1 receptor antagonist has been proven to be, very beneficial, unlike TNF-alpha antagonist, because that is detrimental. So all the rheumatoid, they have no choice. Arthritis patients are given TNF-alpha antagonist and other drugs which are in the combination of the DMA Ds. And Aetna is trying and establishing and validating the use of IL-1 receptor antagonist in many other disease conditions. If it comes, it's going to be a real advantage for our elderly. So what it does is it increases the anti-inflammatory status and decreases the pro-inflammatory status, and there's a synergistic response with TNF-alpha blockers, IL-1 alpha blockers, and those are some IL-6 blockers and B-cells and T6 blockers. You guys must know how that DMARD is and what it does. And then you have vaccines, of course, and you need to take periodic vaccinations. Every 5 years you have to have the vaccine given. And if possible, start from 50 itself. I gave a seminar in nursing school last year in pharmacological upgrade, and there was a patient, she got up and she said she is 65, but her doctor doesn't, she had one myocardial MI attack, and she is not given pneumonia vaccine. So she is asking and requesting the doctor, but the doctor is not giving for some reason. So she was telling, please can you help? I said, I'm not a doctor. She said, why are you not a doctor? You should have been a doctor. I was not lucky. So the very, very best thing out of all is good exercise, social activities, and a healthy diet, which can keep you really, really safe. And with that, I would come to my personal research interest, because I've shown you what we are doing, but how I'm going to take it is I want to use these parameters to reduce aging in the first place, or reduce aging-associated disorders. And I want to see the impact of these changes on epithelial cells, macrophage function, and how the epithelial mesenchymal transition occurs, and or it is blocked. And I want to use, with an infection model or a disease model, I want to use ubiquitination strategies, stress-induced apoptosis and chaperones, to see how we can combat with this situation if it is already established. And then the beautiful part of the next idea is my AHA grant, which I have submitted to AHA for a faculty transition grant. And this grant talks about how these endothelial cells carry those bacterial products, and how cardiomyocytes and endothelial cells of the heart are affected by these bacterial products. And the intervention I have given there is vaccination, statin therapy, antioxidants, oxygen therapy, and ARV blockers. So that's the strategy, interventional strategy I have proposed in my AHA grant. So the first part is my R01 grant, and the second part is my AHA grant. So with that, I come to the summary slide. You guys will be very happy. I restate that senescence may contribute to inflammation during aging, and it increases pneumococcal ligands expression and correlates with increased susceptibility to bacteria, bacterial binding. And there is another exaggeration which comes from the impaired innate immune system that will cause or exacerbate the vulnerability of pathogenic assaults during aging. That, and if all you can do is prevent, prevention is better than cure, and if you can prevent through all these measures, you are going to be good. All this work was done in Carlos's lab, and I'm still working with him on generating a cell line, a cancerous cell line that can lead to senescence. It's a very big project, and one single experiment takes 7 months. So I have completed 3 sets. I need to do 2 more sets to have statistically significant data. So I'm doing that, and it's going to be a very good method, or it's a good strategy for even which we can implement in cancer patients. So that's my interest, and with that I can take any questions. Thank you. How many of you slept?
What type of, in your study, what type of free antioxidants do you plan on using?
We want to use fish oil or omega-3 fatty acids in general. We did a pilot experiment on omega-3, and we also wrote an R01 grant, Carlos wrote an R01 grant, but we got cash. So, but for everyone, the question came in nursing school also. I said, fish oil. They said, oh, what is the dose? 1200 milligrams twice a day. That should be good. You don't take vitamin C or anything, it's a bust. You don't have any,
And vitamin E too.
Yeah, vitamin, no. So fish oil is very good, and flaxseed oil is very good. So in general, I can say, if the treatment or the preventive measure should be homeopathic, you have to have very less, and you shouldn't have a purified compound. So kind of crude things are going to really help your system to get adjusted. You are not going to burden it with severe toxicity.
Okay, thank you so much, Kathy. Dr. Shivshankar, thank you.
Pooja.
Shivshankar. Can you send me this?
Oh, you are thinking, well, man,
Which part of Turkey?